Bismillahirrahmanirrahim. I am Dr. Malik Shija from Department of Microbiology, Cohort University of Science and Technology. Today I am here with my online lecture that is on instrumentation and analytical technique. So in techniques, we have to focus on enzyme-linked aminosorbent assay, which is lecture 12. These are the contents of my present lecture, that is definition. Then we will put some light on the history of enzyme-linked aminosorbent assay. Introduction. We will discuss the principle of ELISA, the different types of equipments and regions that are using in the ELISA, and then we have to follow the general procedure for ELISA. So these will be discussed stepwise. Before going to depth, so the definition of ELISA is basically a widely used laboratory technique to identify peptides, proteins, antibodies, and hormones. It is also used in the field of biotechnology and microbiology as well as in medicine as a diagnostic tool. Mainly in ELISA, antibodies and color changes are used to identify target substance. So we can analyze that an analyte such as antibodies or antigens are present in the test solution. So we have to find out the complex of antigen and antibodies in the samples. This is a widely used laboratory technique for detection of antigen or antibody in the sample. Why we call it ELISA, or what ELISA stand for? So ELISA is basically enzyme linked aminosorbent assay. This clearly indicates that antigen or antibody, because both of our interest is absorbed on the plastic surface. So we are going to coat or label the micro well of microplate, which we are going to use in the ELISA. And antigen is recognized by a specific antibody. So this line indicates the immune system or immunology of the ELISA. This antibody is recognized by a secondary antibody. So in most cases, we are using two types of antibodies. One is capture antibody or primary antibody, and the second one is secondary antibody, which is used to recognize the primary antibody. So in this immune system, or in this system, enzyme is linked with the secondary antibody. So in other words, we can say that enzyme linked antibody. Or if we are using to perform, or we are performing direct ELISA, so there is only one antibody is used, primary antibody or capture antibody, or sometimes we are called it capture antibody. So the capture antibody is also linked with the enzyme, so that's why it is known as enzyme linked antibody. And the last one, we are using two substrate which react with enzyme to produce product, usually colors, and chromogenic material are used like a TNB. So now we conclude from these four lines that enzyme linked aminosorbent assay, how the name was derived from different words. We should also put some light on the history of ELISA. Before the advent of ELISA, radioimmunoassay employing radioactively labeled antigen antibodies were used. Radioactivity served as the reporter signal indicating specific antigen or antibody. As a radioimmunoassay posed significant health risks to researchers, alternatives were sought out. In 1960, enzyme linking process was developed by two different teams. In the same period, aminosorbent preparation technique was published by White and Pora. Independent research papers published in 1971 by Peter Perlman and Eva Engvall at University of Sweden, Stockholm University in Sweden, and some other research scientists in the Netherlands produced the knowledge that goes into making ELISA. So we are going to learn that this technique to prepare something like aminosorbent to fix antibody or antigen to the surface of container. So the surface of container means the coating plate. So the conventional ELISA involves usage of chromogenic reporters as substrate to produce color changes to indicate the presence of specific antigen or an analyte. So this was a little word about the history of ELISA. What is the basic principle of ELISA? So ELISA works by coupling antibody or antigen to assay enzyme. The assay combines the specificity of an antibody and sensitivity of assay enzymes to primarily detect antigen through assay antibody or antibodies through assay antigen. The sensitivity and precision of the assay is enhanced by coating the plate with high affinity antibodies. Basically, ELISA is based on key lock mechanism or key lock phenomena. Elucidates high specificity. So, in the key lock mechanism, antigen in the reaction serve as a key and antibody they serve as a lock. So, key fits into the lock. This clearly indicates the specificity of the key and specificity of the lock because not every key fits into the other lock. In some cases, secondary antibody is used which is conjugated or linked with enzyme mostly alkaline phosphatase or horseradish peroxidase responsible for chromogenic reaction. Further quality and quantity can be determined using plate reader. So different sort of equipments are used in the ELISA. So first of all, we will uh, discuss the micro titer plate, which is made up of polystyrene plate, plaster of polystyrene like material containing 96 wells, and these wells are arranged uh, 12 in the columns and 12 wells are present in the column and 8 in the rows, and each well has a capacity of 350 microliter sample reaction. The other equipment we are using in ELISA are different types of multi-channel or single-channel micropipettes. Different types of micropipettes available in the laboratory uh, from 0.2 micron to 1 ml. If we are performing ELISA and using a single channel pipette, and so we will use it again to reduce the or make it easy. So multi-channel pipettes or digital pipettes are also available, which has a potential to with 8 channels we can see here in this graph. And these 8 channel micropipettes has the capacity to adjust it with the specific digits which it will load and unload the exact amount of sample from the container, and using this multiple set of channel, the chances of contamination will be less, and it can also help out in reducing the work scale. In most labs, ELISA plates are washed manually, but now washing devices are also installed in some commercial and research labs, which are very helpful in proper washing while dealing different sort of ELISA. Here in the figure we can see a washing machine, the washing device which is normally used for the washing of ELISA plates after each reaction or incubation. So this is another type of microplate washer which is digital and can wash the each and every well properly. These are in ELISA we are using different sort of regions. So we are using the coating buffer which is a combination of phosphate buffer and sodium chloride, and this both combinations are known as BPS. Then diluting or washing buffer which containing the phosphate, sodium chloride and twinned. So this the whole solution is known as PBST. We are using blocking buffer. So in blocking buffer we are using wine serum, albumin, and we are using some enzymes which is attached or linked with the secondary antibody horseradish peroxidase or maybe alkaline phosphatase. We are using chromogenic substrates TMB, and finally we are going to another solution which is known as stop solution. In stop solution we are using hydrogen sulfate with a low concentration. This is the general procedure of ELISA. First of all, the well of the microtiter palette are loaded or coated with the antigen. So antigen is added to the plate. Then blocking buffer is used to block the remaining binding site of the proteins. The next step is the application or adding of primary antibody or capture antibody. For detection of capture antibody or primary antibody, we are using secondary antibody which is linked with the N enzyme. Uh, so in the last we add substrate TMB is added, and which for and the enzyme convert the reaction into detectable form, and a yellowish color are produced in the positive wells. So this is basically the general procedure for ELISA. So we are using different sort of ELISA. In case of we are using sandwich ELISA, so the wells in this case wells are coated with the primary antibody, then we apply the antigen so it bind with the primary antibody because it match with this primary antibody. For this then we are going to apply the secondary antibody or modified antibody. So now the antigen is sandwiched between the primary antibody as well as in the secondary antibody, and then in the last we apply the TMB or chromogenic substrate, and the enzyme convert the chromogenic substrate into a reproducible color or detectable color, and we can get the information that in which well of the microtiter parade the antigen is present. So this was all about the basic concept or the basic of ELISA, and we will study about the types of ELISA in the coming lectures. And thank you all students for your kind attention. Allah Hafiz.